So, welcome to the last session. As some of you might know, I was actually a neuroimager, but shifted focus to genomics for the past few years. So I'll try to present the essentials for neuroimagers like myself to go into genomics. That's the, there we go, okay. So here's the outline of the talk. I'll first do a quick recap of GWAS to motivate XQTL analysis. QTL stands for quantitative trait locus. And you can think of it as just associating SNP to some phenotype. And X refers to the intermediate phenotype that I'm doing this QTL analysis on. And for this talk, I'll focus on gene expression, DNA methylation, and histone acylation as the intermediate phenotype. And I'll give a simplified introduction to all these biological mechanisms. I'll then talk about the challenges to XQTL analysis and the type of questions that the genomics community are interested in. For example, if I extracted XQTLs from brain tissues, how well do those replicate in blood or other tissues? Also, for the XQTL SNPs, do they affect just one molecular attribute or multiple attributes? In fact, we can go one step further and ask, okay, the effects of SNP on gene expression, is it mediated via some epigenetics mechanisms? And further, we're going to talk about whether certain XQTL are more pronounced in certain cell types. And lastly, given we have all the XQTL, what can we do with it? Can we actually improve GWAS somehow? And hopefully, within the next 40 minutes or so, all the concepts and terms I just mentioned will become clear. All right, so here's let me move this so you guys can hear me better. So here's the Manhattan plot for a really large-scale Alzheimer's disease GWAS. So again, the x-axis are the SNP's position across the genome, and then the y-axis is the negative log 10 p-values of the association between each SNP and a certain phenotype. So in this case, it would be disease status. And typically, we threshold at 5 times 10 to the negative 8, as mentioned before. Now, one of the important observations from all the GWAS studies is that the GWAS hits usually lie in non-protein coding regions. So if it lies in protein coding regions, we know roughly the function based on the protein that the region encodes for. But then if it's in non-coding region, we really have very little idea of what it does. Now, one way around this problem that people often do is to simply map the SNP to the closest gene. So for example, this set of SNPs will be mapped to CR1, this set of SNP will be mapped to BIM1. And you'll notice that rarely will we just have one SNP being significant. Instead, you'll have like a batch of SNPs that are spatially close to each other being significant together. And this due to something called linkage disequilibrium, as Jason mentioned. And it's just a fancy way of saying correlation. All right. Now, another way to map the SNPs to gene is based on how well these SNPs explain gene expression. And I'll define what gene expression is very shortly. But if I were to map the SNPs based on gene expression, I will find some genes that are actually very far away from the GWAS hits, but yet functionally relevant. And this brings me to the topic of this talk, XQTL analysis. So going from the genome to the phenome, there are actually many interacting biological layers. And when we're doing GWAS, we're sort of skipping all these layers. So it's very hard for us to understand the underlying mechanisms that gave rise to the phenotype. And also, as Jason mentioned, because we're skipping all those complications, usually the effect size is very small, and you need at least like tens of thousands of subjects before you detect any relevant SNPs in GWAS. Now, what we want to do in XQTL analysis instead is to look at the association between SNPs and these intermediate biological layers. So for example, let's say I have a letter change here for a SNP. Would this affect the DNA methylation at this site here, which in turn affects some gene expression downstream. And because these layers are actually closer to the SNP, typically we just need about like several hundred subjects, and we're already able to detect those XQTLs. But before I go into XQTL analysis, let's define some terms and let's talk about what these biological layers are. So here's a simplified picture of a cell. And for this talk, I'll focus on the cell nucleus. But the overall picture here is that you have your DNA, and then you have something called the RNA polymerase, enzyme, that makes complementary copies of the DNA. This process itself is called transcription, and the copies themselves are called messenger RNAs. And then after transcription, the messenger RNAs will leave the cell nucleus, enter the cytoplasm, and then something called the ribosome will attach to these messenger RNAs, use the information encoded by the messenger RNAs to assemble amino acid into proteins. And proteins are what make up our body. And this process here is called translation. And the overall process going from transcription to translation is called gene expression. And as you can imagine, if anything goes wrong along the way, then you might have some protein missing or some abnormal protein being formed. Both cases could potentially lead to disease. Now, there are many technologies for measuring translation, but for this talk, I'll focus on transcription. All right. So here's a picture of a cell again. We have our nucleus, we have our cytoplasm, and then inside the nucleus, we have 23 pairs of chromosomes. Each chromosome is just a long stretch of letters, GC, and then certain stretch of it is called gene, and most of these codes for proteins. Now, within the gene, there are many regions. This position here is called the transcription starting site, and as the name implies, this is the site where transcription starts. And then this stretch here is called the gene body. We have our exons, which codes for the protein, and we have our introns, which are the non-coding parts. And some people believe there's some regulatory functions for these introns, and I'll get back to this point. Now, just immediately upstream from the transcription starting sites is the promoter, and this is where certain proteins called transcription factors binds to initiate transcription. And a bit further away from the transcription starting sites are enhancers, which are also for transcription initiation. All right. So what is transcription? Well, it's summarized in this picture here. And for simplification, usually people draw it as a line, the genome as a line, but in actual, we should be thinking in 3D. And there are many loops there. So for instance, due to this chromatin looping, the enhancer is actually a lot closer to the transcription starting site in true physical distance. And for transcription to happen, first, there's something called transcription factors that binds to the enhancer as well as the promoter. And there are also something called mediator proteins that also binds here to form a complex. And then once this complex is formed, then the enzyme RNA polymerase 2 will attach to the promoter, and then it will move along the DNA to make complementary copies of it. And those are what we call the pre-messenger RNAs. All right. So once you have the pre-messenger RNAs, a process called splicing occurs, which takes out these non-coding regions. And this is partly why people believe the intron might have some regulatory mechanisms, because it could potentially affect splicing. All right. So after splicing, you'll have your mature mRNA, or just mRNA in short, and then these get translated into proteins. And important to note is that these proteins, some of them might be transcription factor, so they will actually loop back and actually like attach to these enhancer promoters, which regulates other genes. All right. And as you can imagine, depending on the location at which a letter change occurs, so for example, if a letter change occurs here, then you might have like some missing proteins or some abnormal proteins. And then again, we call that, it might be transcription factors, or it could affect other genes. And then if a letter change happens here, then maybe transcription factors cannot even bind in the first place. Now, there are actually other mechanisms that could affect gene expression. And what I'm showing here, actually, moving back a little bit, the reason why we know this is that in an individual, all the cells, more or less, have the same DNA, more or less. Because of your recent findings, I can't say exactly the same, but more or less the same. But then some cell becomes like parts of your brain, some cells become part of your heart, etc., right? So there must be something beyond the genome that regulates whether a certain gene gets expressed or not. And depending whether the gene gets expressed, that you might get brain cells, you might get heart cells, etc. So this is what people refer to as the epigenome, so above the genome. And as you can see here, the DNA, certain parts of the DNA are like tightly wrapped around these things called histones, and certain parts are actually quite accessible. And as you can imagine, if you have a transcription factor, I want to bind to your DNA, if I'm tightly bound against the histone, it's very hard for the transcription factor to bind there. But then if it's open, then it's a lot easier. So it turns out that if the tail of a histone gets acylated, so all these are just jargons, but gets acylated, this complex structure will actually relax. And then your transcription factor can bind, and this tends to increase gene expression. And this process is called histone acylation. Now, another epigenetic mechanism that is very important and very widely studied is DNA methylation. And what this means is that there's a methyl group that binds to the CpG sites, and CpG sites is just a fancy way to say a position on the genome where the letter C is followed by the letter G. Right. And if the binding happens on the promoter, then there's usually that would repress the gene expression. And there are like two reasons for this. First of all, the methyl group itself might be blocking the transcription factor physically. And the other reason is that usually when you have DNA methylation on the promoter, it will recruit some other proteins that tends to cause the chromatin to become compact. And then therefore, again, the transcription factor is harder to bind there. Now, if DNA methylation happens on the gene body, then no one knows what will happen. Empirically, it's been observed that it tends to increase gene expression, but yeah, not a lot of evidence on that. All right, so quickly recapping, we have, I'll talk about like two key components of a cell. We have our cell nucleus, we have our cytoplasm. Within the nucleus, we have 23 pairs of chromosomes. Each one is a long stretch of letters. Certain regions are called gene. So within the gene, we have our exon, we have our intron, we have promoters and enhancer for transcription initiation. And we talked about transcription, where RNA polymerase makes complementary copies of the DNA. And when people measure gene expression, usually they're counting the number of these mRNAs using a technology called RNA-seq. Now we also talk about the epigenome. So we talk about DNA methylation, which tends to silence a gene if it's on the promoter. And people usually use something called the Illumina 450K for measuring this. And furthermore, we talked about histone acylation, which tends to increase gene expression, and people use ChIP-seq for measuring this. Now, there are many other technologies for measuring, for example, how accessible the chromatin is. But for this talk, I'll focus on those three technologies. So the RNA-seq for measuring gene expression, Illumina 450K for measuring DNA methylation, and the ChIP-seq for measuring histone acylation. So in particular, the dataset that I've been working with is from the ROSMap consortium. This dataset is actually one of the largest multi-omic dataset for human brain tissues. And within this dataset, we have gene expression data, DNA methylation data, histone acylation data, and actually we have other omics data not shown here. And what we want to do is to determine where, so for example, let's say a little change here, which molecular attributes would be affected. And that's pretty much what XQTL analysis refers to. So you can think of it as just correlating a SNP to some molecular attributes. And if this association is significant, we call that pair XQTL. But the question is it as simple as that? Well, the first challenge is the high dimensionality. So after you do some standard quality checks, you'll get about 7 million or more of SNPs, several hundred thousands of methylation probes, several tens of thousands of histone acylation peaks, and highly expressed genes. So if I were to do all pairwise association tests, then we're talking about several trillion tests. So even if you have the computational power, you have your patience, and you have no statistical power at all to detect anything. Now, the fortunate part is that in past blood-based studies, what people observe is that the number of eQTLs drops drastically with respect to the distance between your transcription starting site and the SNP. So notice that after about 100 kilobases, there's not too many eQTLs left. So if I use a window of 1 megabase, then I should be able to cover majority of the eQTLs. And people observe a similar trend for histone acylation QTLs. And as for methylation, people observe you only need about like tens of kilobases. And if we apply those windows, then we're down to several millions of association tests as opposed to trillions. Now, the next challenge is confounds. For things like sex, age, batch, etc., we have measurements for. But in genomics, there are certain things that are much less accessible, in particular, cell type proportions. So what this means is this. If I get a sample of brain tissues, inside that sample, I'll have neurons, I'll have microglia, astrocytes, etc. And depending on the relative proportions of these cell types, certain genes will be more expressed than others. But this is solely an artifact of, for example, how much neurons I have in the brain tissues. The fortunate part is that in past studies, it has been observed that the first few principal components of your molecular data actually highly correlates with the confounds, as partly shown here. And there's strong evidence showing that the first few principal components actually highly correlates with cell type proportions. So therefore, we can use PCs to account for various confounds. And as for the number of PCs, one way of choosing it would be to look at how many QTLs, XQTLs you detect with respect to the number of PC you include. And when I did this exercise, roughly 10 PC will do the trick. All right. Now, the last challenge related to the high dimensionality is multiple testing. So just a very quick recap. If I were to threshold at alpha, then the probability of not falsely rejecting one hypothesis would be 1 minus alpha. If I have n hypothesis, then it would be 1 minus alpha raised to the n, assuming independent hypothesis. And so the probability of making one or more error would be 1 minus this probability, which people refer to as the family-wise error rate. And if you flip the equation, then you get your Bonferroni correction threshold. Now, Bonferroni correction would be super stringent in this case because we have so much correlation between the SNPs, as shown before in the Manhattan plot. You can use like FDR, but if you're using FDR, make sure you use the version that accounts for arbitrary dependencies. And often in the literature, people don't like clarify that. And it makes a major difference. Because as you can see, like the p-values can be like exactly the same all across, and then yeah, you'll just like get a lot of false discoveries. All right, and why do I care about the threshold? That's the part of the reason. So several years ago, people put a dead cell in the scanner and yeah, found some brain activity. And what happened was that they did not correct for multiple comparison, as simple as that. And if we don't correct for multiple comparison, there are other interesting findings. For example, the amount that US spent on science related to suicide, or the number of people drowned related to the number of films that Nicolas Cage appeared in, as well as how many like PhD awards we get depending on how much cheese you eat. And you can find more important findings at this website. All right, now back to XQTL analysis. So here's the pipeline. First, for all pairs of associations, we apply a window to reduce the number of associations to be tested. And then for each molecular attribute, we remove the effects of confounds. And then we apply spearmint correlation between the denoise molecular feature and the SNPs that are within the window. And then after that, remember to do bond-forming correction or some kind of correction. All right, so after you get your XQTL, one of the first things that you should do is replication. And at a very high level, what this means is that for the relevant associations that are found in one experiment, how well does that overlap for the relevant association in another experiment? And for this, I used something called Pi1 statistics. And the idea, actually, backing up, what this does is that it gives you an approximation of the proportion of non-nulls based on your p-value distribution or the shape of it. And the idea is this. If all my associations are nulls, right, then my p-value distribution should be just a uniform distribution. But if there are some non-nulls, then I'll be left skewed like this. And the idea is that if for, let's say, p-value lambda of 0.6, if everything is null, I know how many associations should be null, right? So if I use lambda of 0.6, then it will be 0.4 times n being the number of associations being tested. So what Pi1 statistics do is that it computes the empirical number of p-values that's greater than 0.6, and then dividing that by the theoretical number. And then 1 minus that will be what people call Pi1 statistics. But of course, depending on the lambda that you choose, you might get a different Pi1. So what people do is that they use all different lambdas and then fit a spline through it and then look at the value at lambda equals to 1. All right. So here are the replication results. What I did here is that I took a whole bunch of XQTLs from various studies, and then I look at the p-value distribution of these XQTLs in the ROS-map data. And it's reassuring to see that, it's reassuring to see that it replicates well for brain studies because the ROS-map data is extracted from brain tissues. It does reasonably well for blood, likely due to common immune functions, but not replicating well for some irrelevant tissues, which is a good sign. Now, although there's high replication between brain and blood, if we were to plot the p-values for brain and p-value for blood, you'll notice that there are certainly associations where you have a super low p-value for brain and not for blood, and vice versa. So there's definitely some EQTLs that are brain specific, meaning we cannot simply just say draw blood and then hope that it will get us all the brain XQTLs. All right. And among the XQTL SNPs that were found, so there's several hundred thousands of them, about tens of thousands of them actually affects multiple molecular traits. And these might be particularly important since it affects more things. And in fact, we can take one step further and ask whether, for example, the effect of SNPs on gene expression, whether if it's mediated via some epigenetics markers. And for this, I use something called a causal inference test. But due to time, I think I'll skip the math behind this. But one thing to know is that in this test, you need to test an alternative hypothesis where you're checking for beta equals to 0. And this is the tricky part for this causal inference test. So look at the notes later on of how to do this. And when I apply this to the ROS-map data, what we observe is that for most parts, the SNPs actually directly affects the gene expression as well as the epigenetic marks, but there are some particular cases where full mediation is observed. All right. And then one more question that's particularly relevant for follow-up studies is that are certain XQTLs more pronounced in certain cell types? And for this, what I did was, again, I'm testing for expression here as the response, and I'm modeling that with the main effects of SNPs, but in addition, I put in the main effects of cell type proportion as well as their interaction, and this is the term of interest. So basically what this means is that for the different genotype, does the effect of cell type proportion affect gene expression differently? And for estimating the cell type proportion, one way to do it is to use gene expression markers as summarized here. But unfortunately, this actually did not do too well. So even at a very relaxed threshold of FDR of 0.2, we only got like very few cell-specific XQTLs. And this is actually a very active area of research where people are doing single-cell studies, etc., to determine which genes can be used as markers for different cell types. And in the interest of time, I'll skip this, but the point here is that your gene expression, as well as all those epigenetics, it captures not just cell proportions, but a lot more. It actually captures a lot of things that you're exposed to. So you can use this as a surrogate to model the environment, and this is one of the areas that I'm working on currently. All right, and then final thing is, what can we do with all these XQTLs? Well, one observation in the past is that many of the GWAS hits actually are XQTL SNPs. So the easy way would be to just restrict your GWAS analysis to the XQTL SNPs. Therefore, you can reduce the number of tests. But the problem is, what if the GWAS hits are not XQTLs, or we just did not have enough sample size to detect all the XQTLs? Well, one way around the problem is to use something called weighted GWAS. And the theory is that if you have the p-values from a GWAS, you can weight the p-values, and if the weights are greater than 0 and sums to 1, applying bound-forming correction to this, you're guaranteed that the family-wise error rate is still controlled. So the idea now would be to upweight your XQTL SNPs and downweight the other SNPs. And I have actually done this exercise to many GWAS studies, one example shown here, where I found 18 additional loci. Right. And in fact, you can go one step further. You can take your XQTLs and then use that to generate a weighted sum of the SNPs set scores. So the idea here is that first, you use your genomics data to build a model, so you have a weighting for the SNPs, and then you take the set score off from the GWAS studies, and then you can do a weighted combinations of them. And the way that this can tie into imaging genetics is that you can take the set scores from the imaging genetic study, and then just apply this model. Then you can get gene level interpretation as well as interpretation at the epigenome level. All right. And all these results you can find in this paper, and I've also uploaded all these XQTL, etc. on this website. And there's an interface that we made where you can put in a SNP, and that will tell you which molecular attributes is associated with. All right. So here's some of the questions. First question is, what are the different regions of a gene and functions? Anyone? Skip that? No, that's pretty much it. That's okay. So just acknowledgements. The data is actually from these two principal investigators. She's my boss, and we're hiring. So if you guys are interested in like genomics and mixing that with imaging, let me know.